Somewhere in the open Pacific, a shape descends through the last column of fading blue light and enters the dark. The animal is 18 meters long. Its head alone accounts for 1/3 of its body, a blunt rectangular mass unlike anything else in the ocean. As it passes 300 meters, the pressure around it rises to 30 times that of the surface. Its ribcage begins to fold inward. Its lungs compress. Its heart rate drops to a fraction of its resting rhythm, and yet it continues to descend, past 600 meters, past 1,000, past 2,000, in absolute silence, following sounds it is generating that no human ear could detect, toward prey no human eye has ever observed alive in its natural depth. It will stay down for over an hour. Then it will surface, breathe, and do it again. Today, we're diving into why nothing about the sperm whale is normal. Not its origin, not its anatomy, not the organ that occupies its skull, not the way it breathes, dives, communicates, or organizes its society. At every level of examination, from its evolutionary history to the molecular structure of its muscles, the sperm whale does not behave the way biology expects an animal of its kind to behave. It exceeds every framework that science initially placed around it. 50 million years ago, there was no sperm whale. There was no whale at all. What existed instead, on the riverbanks and floodplains of what is now Pakistan and India, was a wolf-sized four-legged mammal called Pakicetus. It had hooves. Its teeth were suited for catching fish. Its closest living relatives were even-toed ungulates, the group that today includes deer, pigs, and hippopotamuses. Pakicetus did not look like the ancestor of the largest toothed predator on Earth. But it was. Over the following 15 million years, the descendants of Pakicetus progressively shifted their lives toward water. Limbs shortened, nostrils migrated toward the top of the skull. The pelvis became vestigial. The body stretched and widened. Each transition was not a dramatic leap, but a slow accumulation of heritable changes, driven by the ecological pressures of an ocean that offered enormous food resources to any animal capable of reaching them. By 35 million years ago, the lineage was fully aquatic. By the time the ancestors of the modern sperm whale had fully emerged, the land-dwelling origin of the entire group had been architecturally erased, buried beneath layers of biological reinvention so thorough that the whale's terrestrial past survives only in vestigial hip bones, tucked within the body wall, attached to nothing, serving no function, the last remnant of a skeleton that once walked on ground. The sperm whale genome, sequenced and analyzed in recent years, carries the molecular record of this transformation. Researchers identified specific genetic adaptations, alterations in genes governing protein function, cellular repair, and physiological regulation that distinguish the sperm whale's biology from both its terrestrial ancestors and from other cetaceans. The genome reveals not one dramatic evolutionary event, but a continuous process of refinement, the biological equivalent of a complete architectural renovation carried out over geological time while the structure remained in use. What that renovation ultimately produced is an animal whose proportions challenge immediate comprehension. A fully grown male sperm whale reaches 18 meters and weighs up to 57 metric tons. 1/3 of that length is the head. That head, that enormous, blunt, rectangular head, is not filled primarily with brain or jaw or sensory apparatus in any conventional sense. It is occupied, in large part, by a structure that science is still working to fully explain. The spermaceti organ is unlike anything else in animal biology. It is a barrel-shaped mass of waxy oil, encased within a connective tissue casing, extending the full length of the upper head. In a large male, it measures up to 5 meters in length and holds close to 2,000 liters of spermaceti, a substance so pure and so unusual in its chemical composition that it was, for centuries, mistaken for the reproductive fluid of the whale itself, hence the animal's name. It is not. What it actually is, in functional terms, depends on which scientific hypothesis one examines. The most thoroughly supported function of the spermaceti organ is acoustic. The sperm whale produces sound through a pair of structures at the front of its nasal passage called the phonic lips, narrow muscular folds that generate pulses of compressed air. These pulses travel backward through the spermaceti mass, reflect off a dense air sac situated against the front of the skull, and are redirected forward, focused, and amplified through a fatty lens called the melon and emitted as a directed acoustic beam. The result is the most powerful biological sound ever measured on Earth. At its peak, a sperm whale's click reaches 230 decibels. A pressure wave louder than a gunshot fired directly beside the ear, louder than the takeoff of a commercial jet at close range. This sound is used for echolocation, allowing the whale to map the geometry of the deep ocean in complete darkness, detect prey at distances of hundreds of meters, and navigate across ocean basins with precision that no engineered sonar system fully replicates. A second hypothesis, supported by decades of thermal and physiological analysis, proposes that the spermaceti organ also functions as a buoyancy regulator. Spermaceti oil transitions between liquid and solid phases across a narrow temperature range. By selectively increasing or decreasing blood flow to the organ, the whale may be able to warm or cool the oil, shifting its density and thereby adjusting the whale's buoyancy without expending muscular energy. At depth, a cooled, denser spermaceti mass would allow the whale to achieve neutral buoyancy passively, sinking under its own adjusted weight, preserving oxygen stores for the hunt rather than expending them on swimming. A third hypothesis, supported by structural analysis of male sperm whale skulls and by historical accounts of whale behavior, proposes that in mature males, the spermaceti organ additionally functions as a physical weapon. The architecture of the male forehead is markedly more reinforced than the female's, with denser connective tissue and a structural geometry consistent with absorbing and transmitting impact force. In November 1820, the American whaling vessel Essex was struck twice by a large male sperm whale and sunk in the open Pacific, an event so extraordinary that it became the historical basis for Herman Melville's Moby Dick. Multiple other documented cases of whaling ships being rammed exist in the historical record. Whether this behavior represents intentional use of the head as a weapon in defensive aggression, or whether the spermaceti organ evolved primarily for this purpose in the context of male competition, remains a genuinely unresolved question. What is not disputed is that no other animal on Earth carries a single anatomical structure capable of serving as a precision acoustic instrument, a potential buoyancy control mechanism, and a structural impact weapon simultaneously. The organ does not fit neatly into any single evolutionary category. It may not have been built for any single purpose. The spermaceti organ explains how the sperm whale hunts. The physiology of its body explains how it survives the hunt itself, because what the sperm whale does every day, descending nearly 3,000 meters below the surface on a single breath, should, by the biological standards applied to virtually every other air-breathing vertebrate, be impossible. At the depths, a sperm whale routinely reaches. Water pressure exceeds 300 atmospheres. At such pressure, air-filled spaces in the body become a liability. Nitrogen dissolves into blood under pressure, and if that nitrogen comes out of solution too quickly during ascent, it forms bubbles in the bloodstream and tissue, the condition known to human divers as decompression sickness. Rigid lungs would fracture. Gas-filled sinuses would collapse catastrophically. The laws of physics that govern pressure are indifferent to the size or evolutionary sophistication of the animal subject to them. The sperm whale's body does not resist these forces. It cooperates with them. The rib cage is not fixed, but flexible, constructed to collapse progressively as pressure increases with depth, folding inward around the lungs and compressing them toward a fraction of their surface volume. As the lungs collapse, the air they contain is forced out of the alveoli and into the upper airways and trachea, regions where gas exchange with the blood does not occur, effectively eliminating the nitrogen absorption problem before it begins. The whale descends to 3,000 meters not despite its flexible rib cage, but because of it. Simultaneously, at the onset of a dive, the whale's heart rate drops dramatically, a physiological reflex called the dive bradycardia response, common to all diving mammals but exceptionally pronounced in cetaceans. Blood flow is selectively redirected away from organs not essential during the dive, the kidneys, the digestive system, the liver, and concentrated in the heart, brain, and working muscles. The spleen, normally a reservoir of red blood cells, contracts forcibly and injects an additional surge of oxygen-carrying capacity into the circulation. Every organ system in the body coordinates into a single synchronized response, oriented toward one goal, extending the duration of the dive. The muscles themselves are built differently. Oxygen-binding proteins called myoglobins, which in most mammals constitute a small fraction of muscle mass, are present in sperm whale muscles at concentrations so high that the tissue appears nearly black. Each myoglobin molecule stores one molecule of oxygen. The muscle is, in effect, a biological oxygen tank, a dense, distributed reserve that sustains cellular function long after the lungs have been compressed into silence. The deepest confirmed dive of a sperm whale reached 2,992 meters. The longest confirmed duration was 137 minutes. Both figures surpass the operational limits of most military submarine designs in service at the time the recordings were made. None of this explains why the sperm whale needs a brain that weighs nearly 8 kilograms. The sperm whale possesses the largest brain of any animal that has ever lived on Earth. A mature male's brain averages 7.8 kilograms in mass and exceeds 5 times the volume of a human brain. It is not merely large in absolute terms, it is disproportionately large relative to comparable deep-diving species. Its neural tissue contains structures that were, until their discovery in cetaceans, considered markers of uniquely human or great ape cognitive architecture. Von Economo neurons, elongated spindle-shaped cells associated in human neuroscience with self-awareness, empathy, and the capacity to process complex social information, are present in the sperm whale cortex in significant numbers. They were first identified in humans, then in great apes, then in cetaceans. The taxonomic list of animals in possession of these cells keeps expanding, and with each expansion, the assumption that complex social cognition is a uniquely human or primate phenomenon requires revision. What the sperm whale does with this brain is becoming progressively clear, though the full picture remains incomplete. Communication is carried out through rhythmic sequences of clicks called codas. Each coda is a brief, structured pattern, 4 clicks or 5, arranged at specific intervals with variations in tempo, rhythm, and what researchers describe as ornamentation and rubato. A landmark study published in the journal Nature in 2024, produced through a collaboration between MIT's Computer Science and Artificial Intelligence Laboratory and the Research Initiative Project CETI, demonstrated that sperm whale codas possess a combinatorial structure. Rhythm, tempo, rubato, and ornamentation can each vary independently and be combined in ways that significantly expand the expressive range of the system, analogous to the way phonemes combine in human language to form a vastly larger set of possible words than the phoneme inventory alone would suggest. This is the first documented evidence of a combinatorial coding in a non-human animal communication system. The codas are not uniform across the species. Across the Pacific Ocean, researchers have identified 7 distinct clan groupings, each characterized by a shared coda repertoire, a dialect. Clan membership is not genetic, it is cultural. Young whales learn their clan's coda patterns from their mothers and from other members of their social unit through a process of social transmission that mirrors, in its basic structure, how human children acquire language. Whales that share a dialect associate together. Whales from different clans, even when they share the same ocean region, maintain distinct social identities through their vocal patterns. A 2022 study published in the Proceedings of the National Academy of Sciences proposed that these coda dialects function as symbolic markers of clan identity, the acoustic equivalent of cultural insignia. What we know is that the sperm whale has been present in the oceans for tens of millions of years, and that for the vast majority of that time, the exchanges it conducted in the deep ocean were entirely unheard by any other species.